Hello, everyone. Am I audible? Hello. आवाज नहीं आ रही। मैं हेलो हेलो कर रही हूँ, मुझे आवाज ही नहीं आ रही। ये बंद हो गया फिर से। पॉज क्यों आ गया ये? पॉज आ गया देखो। रिज्यूम रिज्यूम फुल स्क्रीन कर दूं ना? आवाज कुछ तो बोलो, कोई भी आवाज नहीं आ रही थी। अच्छा, good evening everyone over here. Am I audible?
Yes, audible.
Yes, thank you. Thank you. So, myself, Dr. Lippy, I'm as a vice president in Advanced Cell Group. This is a kind research lab 3 in Noida, Delhi. So today, Delhi, this company focus on more on research and and the cell culture as to mainly stem cell culture along with the primary cells. We have one division that is Kosheeka, that is more focused on the primary cell. And this is we are preparing the raw material for further research for the academia or any in vitro drug screening in a pharma company or biotech startups. So my today's topic is to how these cell culture platforms, I mean, the research in cell culture can be good for especially for the pharmaceutical company, because later on we see in the following slides that the pharmaceutical companies, there is always a chance of drug failure, and many, because in recent years, you can see, we show you the graph that is drug failure in the clinical trial. Why? Why we can overcome this. So this is one of the platform we can do, more research required definitely. So we can give the platform to the pharma company so we can reduce the burden of the drug failure during the clinical trial, and the drug efficacy will be more efficient for the treatment of diseases. So we'll start with introduction, why this is needed. So Vickery Healthcare is highly dependent on drug research and development. So it is essential to promote drug innovation, an ongoing process. Traditional drug discovery involves a series stages of development of new drug, and you know that it takes about 15, 20 years to come into the market. And since we have basic researcher established the 2D cell culture, that is in the scientific community, which improved the cell-based drug screening as a very initial stage because it's required. We'll see in the following slide how drug developed with a big series of research and the trial. And the scientific community has improved the cell-based drug screening due to the 3D cell culture, followed by 3D multicellular model to describe the biological complexity better, because 2D has some limitation, and the simulation of the in vivo microenvironment, because 3D is more, the 3D model is more towards the in vivo, which is required for the any drug initial screening, or you can say during the development. High-throughput screening is carried out after target confirmation, follow the optimization of the compound structure, animal testing, and final clinical trial, which is the process of the drug development. And especially for the chronic illness issues, that is the cancer, which is the most therapeutic area, the overall success rate for products in the clinical approximately 10% only. So because we have to understand the reason of the failure, why for this, but they've required certain collaboration of different basic research as well as the clinical research. So objective of today's seminar to give it is the urgent requirement of this basic cell culture platform to initial drug discovery. Sorry, I'm a little bit in a got an urgent call. Hello? Sorry, actually the call from my home. So that is what's the requirement. I mean, it is the requirement of today's, because you can see the development of cancer drug, because lots of failure in this regard. So because we need a 2D, 3D cell culture along with the new platform already rise with this microfluidic, maybe you heard about that, for the development of anti-cancer drug. And that improved the strategy associated with the cell-based methods. So this is the this slide showing during any pharmaceutical companies, during the development of the um, drug discovery, we need a basic research. You can see here, require a basic research, and lots of funds required as well, and then lots of investment. And then preclinical, followed by clinical trials and FDA approval. Initially, in basic research, what we have to do the target identification as along the validations, which require the basic model, how we target, how we identify the right target and do the validation. This is under the good laboratory practice. And you can see here, basic research, we have to do that target identification and target validation. So it's time taken, as well as lots of capital involvement, lots of investment required here. And followed by preclinical development, it's also a huge amount, and followed by clinical development. So you can say 10 to 15 years takes to come into the market. So we have to see at the base level for the development of the drug. So, and you can see this graph, the number of drug approved per $1 billion per R&D. So you can see the declination of this drug approval, and with respect to the money involved in the research. So that's the importance we are now giving to the cell culture. What is cell culture? Very basic, is the process of isolating cell tissue from their natural environment and subsequently growing them in a controlled artificial environment. It may be short-term culture, like primary culture or secondary culture, or established culture, we call it as cell lines. So just a little bit more, little bit about the cell culture history. To start in 19th century by Cynthia Ringer, developed a salt solution containing the chlorides of sodium, potassium, calcium, and magnesium to maintain the beating of animal heart outside the body, followed by other scientists. And also an important one is the 1907 Ross Granville Harrison at John Hopkins University made the first ever cell culture and his technique called hanging drop method. And he also isolated the embryonic nerve fragment and over the glass slip and the lymph vaulted and cover slip inverted on the glass. So this method is called hanging drop method. So I already mentioned that is the importance of cell culture, little bit more about it, discover drug efficacy, delivery, pharmacological actions, cytotoxic also important during the development of the drug. Because the cell-based assay, we can do the bios in the most comprehensive approach to data harvesting quantitative physiological relevant information of drug during the screening process. So this is the basic where we can get the maximum data for our target identification with the little amount of money involved. And you know that we spend about more than 135 billion yearly on the screening of the medicine. So this is an in vitro model that is very economical, will be based assay, accurate represents a system to studies and which express the relevant factor signaling molecule which is required during the screening of the drug. We can get the data. So we'll start with the 2D culture. We already, everybody know about, maybe they are from the background of cell culture or laboratory technologist. They can know about this 2D culture. And I'm just going to highlighting the very basic thing here. That is the because it may be the conventional cell culture commonly, that is what we are growing in the cell is the 2D culture. It is maybe inherent, that is dependent, this is dependent on the rigid surface. It attached to the rigid surface with the mechanical support, which is we call it anchorage dependent cells as a monolayer. And, and they made monolayer cells and which secrete the cellular growth factors and require some nutrient to grow. So till date, cancer therapeutics still carried out 2D because it's very easy and inexpensive and well established as well. But there is some limitation on the 2D culture because it does not represent the real cell environment. Maybe morphology or phenotype cells can be differ. And there is a limitation access of the monolayers as well as the ingredient, maybe gene expressions, and allows for the study only one cell type. So the main limitation here, this 2D model lacks the microenvironment, such as complex geometric architecture, paracline signals from the neighboring cells, machinal property, nutrition, oxygen, to mimic the native tissue, what we have inside the body. So this is lacking in the outside in vitro 2D model. This micro strongly influence the cell behavior and function also, like proliferation, differentiation, or metabolism. And also the cell-based assay, a main hurdle to design a sufficient powerful detection method with adequate signal noise, because there is to maintain the physiology of the cells, because not mimicking the tissue what is we have, that's the signal also not very accurate. Now coming to the 3D culture, it is artificial created environment which the molecular cells are permitted to grow or interact with the surrounding in 3 dimensions. And it's almost mimic the cell natural environment. So why 3D culture? So we have the 2D culture, which is grown a flat surface, um, where the communication signal between the two cells is very minimum and and uh, difficult to target the valid in this cell culture system because there is a monolayers of the cells. While 3D they have attached to one another, form cell to cell attachment. They are, means communication is well uh, improved with as compared to 2D, and it's very complex as well, that mimic the native tissues. So in this case, targeting of validation, the target validation will be maybe optimal during the new drug discovery, and because the multi-layers formed. So why potential of 3D for drug testing? Because maintain the normal morphology as well, and response to 3D is about similar to 2D, but there is stability is there, more stable and longer lifespan as compared to 2D, sorry. Cells in the 3D culture are undisturbed, where cell 2D culture will tryptonize when it is reach the influence. This is also one of the factor in which the cell drug testing is, optimum. So we have the three-dimensional cell culture system. So we have generally, maybe you guys heard about the spheroid and organoids. Organoids are the complex cluster for organ-specific cells. It's the organ-specific cells. Mind it, note it is organ-specific. And like stomach, liver, and bladder, maybe they are made up of stem cells. The progenitor is self-assemble, and only when there is an extracellular involvement. For example, collagen and other matrix. While spheroid is simple, a symbol of the broad range of cells. Don't require for scaffolding to form 3D. They are self-assemble and regenerate. So I have mentioned here some of the organoid which is generated already in the culture techniques. And also there is advantage, disadvantage of this 3D culture. Mentioned that is spheroid is very easy to use it because organoid is organ specific and patient specific. As we need a scaffold as well. And followed by this can be help in making organ chips and 3D bioprinting. So here you can, giving the example of stem cells, this is self-assembly, and this is made of organoid, and this can help in the drug screening or therapeutics later. So this is the basic difference. Already, I think you know about that. We can make it from the cells or stem cells both. And this is the already research is going on to make the brain cells, organoids, intestine, liver to work, do research the better way as compared to previous 2D monolayer cell culture. And also important is the toxicity screening also, because during drug testing, we need to do the toxicity. So we need a proper cell-based system. So here, one of the hepatocyte, HepG2, heparg cell, which is required for the liver toxicity test. So a little more about the 3D culture, which can be scaffold-based and which is non-scaffold-based. Scaffold-based because polymeric, different kinds of scaffold, biological scaffold, and other kind of surface microplates. This is patterned one. And this non-scaffold, that is the hanging drop cells. And one of the new technology coming out of the microfuel, we'll discuss a little more about that. And the bioreactors and gels. And this is about the polymeric hard square. I'm not going detail. Maybe you can get it from any literature. This is the more detail. This is just and this explaining you the, because 3D requires a scaffold, so different kind of scaffold already there. Um, like matrix of hydrogels and also liquid matrix, collagen, that is a biological scaffold, hyaluronic acid, cellulose, some synthetic scaffold also get polyethylene glycol, polyvinyl alcohol. This is another kind of scaffold both because that is the patterned surface microplate. It is slit in the pattern and square, and then just create, which is usually coated to create a low adhesion surface. Bottom is thin and transparent for microscopic imaging. And this is used for the drug testing and cell biological analysis at the molecular level as well. And the biological, that is from the natural for biological origin, as you know, is about fibronectin coated plates as well, also available collagen, laminin, gelatin, and a cell can attach easily, and you can do the 3D culture with there. And this is the hanging drop and spheroid microplate also available. And bioreactor where is also very common. Maybe you heard about that, where in the antibody production we use this one. Also in the stem cell culture, I mean, scale up production, they require bioreactors. So there may be the spinner flask kind of perfusion bioreactor, which is combination with this scaffold. Is all there. And gel, which is the soft tissue-like stiffness that mimic the extracellular matrix. And this is the natural source, and which is maybe similar to collagen and alginate. Commonly used these days is matrigel, and synthetic one is polyethylene glycol, which is also called hydrogel in research. Because it's high viscous, sometimes very difficult to use. So we have to standardize the process. So some basic few things I want to highlight here, that is, which is considered when doing any cell type assay or cell-based assay for any testing or the drug testing or whatever. So for example, when you select the assay, so what kind of cells require for your assay? That must be amenable, faithfully represent your system, and necessary factors a signaling intermediate must express. This is the important things while choosing the which cell system we have to use while doing depending upon your experiments or research. For example, I'm giving here, suppose you design assay for cell viability assay. So we generally use a metabolite ATP. So we need to remember the cell line generally have a cell lines, which is having higher metabolic rate than primary cells. And similarly, primary cells have higher spontaneous rate of death as compared to cell lines. So we have to say, while we have to think before doing the cytotoxicity test or viability, say what cell lines or what required for my purpose. That is, it's very important things. Other factor which can influence your assay, that is the treatment choice, your duration, concentrations, assay incubation time, what the seeding density of the cells. Passage numbers, this is very important for their assay. Because different cell types have different susceptibility to injury or test compounds that are toxic or cause apoptosis. So different, their level of different susceptibility. So we have to see also under consideration. And the same agent can induce apoptosis in one cell line may have no effect on different cell lines. So this is also very important. So comparison of 2D and 3D culture, this all everybody know, just highlighting few points here. And This is 2D is now very not mimicking the natural structure of the tissue or the heat mass, but 3D is definitely mimicking the 3D form, which is required for the any further drug screening in initial stage. Characteristic of 3D culture methods, we are maybe suspension. Single cells are seated in non-adherent plates, and advantage is simple, easy. And some cell line needs expensive plates coated, because collagen coated or fibronectin, so they're a little expensive. Maybe some are gel based. And some cultures on scaffold for the culture. There is different characteristics. So I have given some example here, that is some of the commonly used cell line with different compound selections. Like HeLa cell is very important. Screening the active compounds. Already listed 157, or maybe more than, just from the literature. And embryonic stem cells, I think you're aware of this, and it's also seeded, so helpful for the drug screening. And HIP-G2 and HeLa cells, both maximum cell lines are using for the drug screening these days. So it's this next generation culture technology that we are talking about, the microfluidic technology, that technology that mimics the blood vessels in your body, is defined as a controlled, manipulated flow of fluid through the micro-sized channels. Because it is integrated with the 3D culture, ensure your steady blood supply, providing the tissue with a constant supply of oxygen, nutrients, and withdrawal of the waste. So just mimicking in your body vein or artery, going to the tissue and waste product outside the body. So this similar way, and this is this technique is also called lab-on-chip technology. That is very useful for drug testing and economical and further testing, especially for anti-cancer drug development. So what is microfluidics? It's deal with the manipulating and controlling the fluid inside the micro-sized channel. Volume of the liquid usually range of microliters to picoliters, so it's very less volume used. I'm not going much detail, just giving the brief idea because it's more technical here. And and this is forming the, this is the microfluidic chip is set of micro channel with the model into material like glass, silicon, or polymer. And these are connected to in order to achieve the desired features such as mix, pump, control, that is the control system. To achieve that, what we require, the constant supply of fluid to each part, the compartment. So this is developed in early 2000. They are using the polymers, PDMS, for such material. And you can see here, this is the design, the 3D culture included in the microfluidic technology. You can see here, there's a different kind of 2D also can be platform of microfluidic technology can be used, but mainly 3D is better. And there are different kinds of microfluidics, gradient-based, droplet-based, or 3D bioprinter, printed-based microfluidics. So already different kinds of microfluidic already developed here, but in research phase. And there's a different components of the device. That is this is the microfluidic device. This is sample volume, electroosmotic pump, distributor, and the pressure chamber, loading, injection. Everything is there here. And you can see how it can be detect the cytotoxicity because you see the concentration, the color of the drug when it is inside testing onto the cells, and you can see there is MEF cells plated here, and the drug has been tested. So you can see the different concentration and their toxicity level with this. An organ check is the especially the more advanced than lab on chip, that different organ like kidney, skin, liver is going on for their to checking the drug there for the inflammation and the organ specific. It's very helpful for further in drug screening. And this here, you can see the lung chips, and you can see everything is there. This is airflow, blood flow, and other capillaries. So it is easy to detect the cytotoxic level or different efficacy of the drug initial stage screening. And you see that this is a chip. And this is the first already reported in the literature, is this already validated lung chips. And thus I told you that this can be useful tool for the drug screening, because also lab-on-chip technology precisely control, and it's manipulate the some millimeter scale fluids in the tree. And it's very economical, but we need the more validation on this and more data on this for drug screening. Here are different kinds of you say microfluidics apply for overcome the challenges and the physiochemical operation consideration. So it's a big list. I'm not going to detail. So we're just summarizing the webinar. Cell culture commonly used in genetic biological cancer research. They mimic the in vivo conditions. And we talk about the 2D, 3D cultures, how it is combined with the microfluidic technology, help for the drug screening to get the good drug candidates. So later on, we cannot waste money due to failure. And this will be, and this can be fully understand the efficacy, toxicity of the drug. Definitely, preparation of microfluidic is still primarily independent of the soft lithotripsy, how it made, like different kinds of material, raw material, the specific infrastructures, money involved, time consuming. This is the drawback of this, but still research is going on, so we overcome all these limitations. So take home message is definitely there is this limitation of 2D, but still 2D is more acceptable, but we cannot ignore the limitation of 2D. So 3D is more better than the pharmaceutical research, because it's mimicking more your tissue environment, what is in your body. And the use of microfluidic-based 2D and 3D cell culture that better mimic the in vivo microenvironment and dynamic fluid flow condition enable access to mechanism of physiological phenomena that would otherwise become unnoticed. And the reproducible and quicker screening of drug at early stage of development can actively contribute to reducing the late stage failure risk. So you have to think about this way. So we can reduce the later on burden of failure while early stage development through this new technology with the cell culture platform with microfluidic or 3D culture, minimum of 3D culture, because microfluidic still in advanced stage, but 3D culture definitely helps you to reduce the risk of late stage failure of drug. Thank you. I told you we have a Kosheeka website where we are producing primary cells as well as stem cells for industry and the academia, so they can expedite their research, just getting the cell from us. So you can visit our website, kosheeka.com, or any information, or you need any help, technical help, you can write to us info@kosheeka.com. Now I can take the questions. Please, if you have. So I would say you must visit kosheeka.com for more technical support and also experimental support. You should visit our website for primary stem cell as well as the primary cell of different organs. Thank you very much.